Hello guys, so we're gonna learn about an experiment that was performed by Bernard Davis to show that the cell-to-cell connection is necessary for the bacterial conjugation. So let's get started. So he prepared a glass tube and fused them at the base. Okay, so it finally formed a U-shaped structure. Okay, and that U-shaped structure he filled with the nutrient media. Okay, and in the nutrient media he inoculated two types of auxotrophic strains. Okay, so auxotrophic means the organism which lacks to produce certain nutrients and produce certain nutrients. Okay, so in this experiment the two strains of bacteria, one strain of bacteria produces leucine and thymine and lacks methionine and theronine. Okay, and in the second bacterial strain the bacteria produces methionine and theronine. Okay, and they do not produce leucine and thymine. Okay, so basically the if the cell to cell conjugation cell to cell contact is necessary for the conjugation, the result of that it would be that the recombinant bacteria would produce all the 6 amino acids, all the 8 amino acids, sorry. Okay, so all the 8 amino acids will be produced by the recombinant bacteria. But in this experiment that I'm giving just an overview that he doesn't allow two bacteria mix. Okay, so if that or mixing, so how can they produce recombinant cell? Okay, so by giving them not to mix, they can't form conjugation between them. Okay, so it's obvious that there will be no recombination, and then therefore there will be no production of all the types of amino acids. Okay, so in this experiment, like, he proves that only. Okay, so he basically does that at the fusion point of the two halves of the U-shaped glass tube, he inserted one filtered glass filter. Okay, so the glass filter allows the nutrient media only to flow. Okay, and nutrient media will be flow by the pressure created by pressure succession. Okay, you can see on the upper side. Okay, so what happens is it creates pressure so that the nutrient media can move along the both the halves. Okay, so nutrient media are allowed to mix with each other, but bacteria are not allowed to mix with each other. Okay, so as a result, we already know that if the bacteria were not given chance to mix with each other, they can't conjugate with each other. Okay, so the possibility of formation of recombination is almost impossible. Okay, so this experiment indirectly proves that if the bacteria are not allowed to mix with each other, okay, as they are not allowed to conjugate with each other, they can't produce recombinants. Okay, so by that it also means that it's necessary to bacteria to mix with each other to produce recombinant bacterial strain that produce all the nutrients. Okay, so this was the basic experiment. I hope you understand, because this is easy experiment. Okay, so there is nothing rocket science in it.
この想いを消してしまうにはまだ人生長いでしょう